I'm Matt Sandoval. I'm a second-year PhD student in the Anatomy and Neurobiology department in the Diaz Alonso lab, and I'm a T32 fellow through the Center for the Neurobiology of Learning and Memory. My research focuses on specifically two what are considered synaptic adhesion molecules, MDJ1 and MDJ2, and they've both been associated with neuropsychiatric disorders. So I'm sort of studying the fundamental mechanisms that and function of these proteins at synapses, or the connections between neurons, and how those changes and those functions over time during development lead to or prevent neuropsychiatric disorders. I think for me, the biggest part of the T32 training has been the two classes that I've taken as part of the training. They've exponentially expanded the scope of my research, coming from a much more molecular sort of electrophysiological background. Being able to look at the brain and a more network and even computational lens has been definitely mind-blowing, and has added a lot of things to my research and a lot of different research questions that I have and want to pursue. And trying to understand how proteins at the synaptic level might change network or circuit level functions across the brain, and specifically in learning and memory. Outside the lab, I very much enjoy live music, so I go to a lot of concerts, a lot of great local concert venues. I like to hang out with my dog. I also love to watch recorded neuroscience lectures on YouTube. I just love hanging out with friends and visiting local spots for trivia or beer.
I'm Javier Diaz Alonso. I'm an assistant professor in anatomy and neurobiology in the School of Medicine in UC Irvine, and a fellow of the Center for the Neurobiology of Learning and Memory. The research in our lab is focused on synapses. And synapses are the anatomic specializations where neurons contact with each other, and that's where neurons communicate with each other. So research in our lab tries to elucidate the mechanisms that underlie the dynamic organization of synapses. Neurons communicate with each other, and that communication is not always stable. So the strength of synapses is regulated. It can be, increased. Synapses can strengthen, and they can also weaken. And we know that this strengthening and weakening of synapses is important for processes such as learning and memory. One of the projects that we study in the lab is how synapses are formed and how they are restructured during development and throughout the process, live and under the influence of several stimuli and learning mechanisms and processes and things like that. The project that Matt Sandoval does in the lab revolves around one of the synaptic adhesion molecules, which are proteins that are involved in precisely establishing these connections between the presynapse and the postsynapse. These proteins are called MDGAs, and they are super important. So mutations in these proteins can lead to several psychiatric disorders such as schizophrenia and autism spectrum disorder. What Matt is doing is using an array of different approaches, including behavior, electrophysiology, biochemistry, molecular biology approaches, and different surgeries to elucidate the role that these proteins play in the regulation of synaptic communication and synaptic plasticity. Matt actually joined the lab as a lab tech in 2021. He's a truly gifted researcher. He's really amazing and has very skilled hands. But I think much more important than that is that he pours himself into the projects that he does. And he's just such a good team player. He actually started doing things that I couldn't teach him how to do. So he went and learned how to do super resolution microscopy, for example. Now he taught several lab members and even members from other labs in the environment. He's very involved in advocacy and in science communication. Being a T32 fellow is definitely one of the highlights of Matt's experience and a great, gift for us in the lab.